Good afternoon. To continue our program, we'll have the next lecture by someone who was, until very shortly, I only knew him by his many publications and writing about the beautiful plants of South and Middle America. And I'm very glad to have him here and have met him. And he's here to talk about the species which hasn't been much given attention in conferences up until now, Genlisea. Please welcome Fernando Rivadavia.
Good afternoon, everyone. So, I'd like to start off thanking Carnivorous, ICPS, for inviting me. It's a pleasure, it's an honor, it's great to see everybody. Not every day that I get to participate in one of these huge meetings, get to meet so many of you who I know through the forums or emails. So yeah, so I have a very difficult task ahead of me here. Actually, before we start, do we have any Spanish people in the crowd? No, so I'll go ahead and congratulate our Dutch hosts for their second place in the World Cup. I admit I was cheering for Holland, especially because you guys eliminated Brazil, so at least we would be able to say that we lost to the world champions, but oh well. So, I have a very difficult task ahead of me here. I'm going to have to try to make Genlisea sound interesting to you all, especially after lunch, last presentation. Anyways, let's see, where did I put that remote control? Here we go. So, Genlisea, not a very big group, but lately a lot of information has come out about this group. We could basically say that Genlisea has come out of the closet. In the past 10, 15 years, our knowledge of this group has tripled, quadrupled, and this is a genus which we've known for about 200 years almost. So, this boring little group of plants, only about 20 species until recently, known for about 200 years, took a long time to even confirm that it was carnivorous. Basically, as you know, Genlisea is one of the 3 genera in the family Lentibulariaceae. We also have Pinguicula, Utricularia, and there's Genlisea. So, the leaves. They're usually little compact rosettes, similar to Pinguicula and Utricularia. Not all Utricularia, of course. Some of them are a little more compact, some of them a little bit less. Some of them very compact, forming dense gelatinous mats over the leaves. Some of them are not very dense at all, just a few little sparse leaves, not forming a rosette. Some of them have very loose rosettes, and you can even see the traps on the surface of the ground sometimes, depending on the species. And sometimes, in the case of Genlisea repens, there is one species that actually forms a stolon, a creeping stolon like Utricularia. Now, Genlisea is very similar in some ways to Utricularia, in some ways more to Pinguicula. So, for example, it has 5 sepals, like Pinguicula. Okay, Utricularia only has 2 little sepals in the back. It's a very easy way to pick them out in the field. If they have flowers, you're not sure, a little yellow flower. There's tons of yellow flowered species. Pick up the flower, look at the back. 5 sepals Genlisea, 2 sepals, that's Utricularia. The flowers are also very similar. They've got those upper and lower lips with the spurs. So there's one group of Genlisea which have that typical, it almost looks like a Utricularia. Again, if you don't look at the sepals, you could easily mistake these for Utricularia, right? Got that long little spur, upper and lower lip, snapdragon type flower. And we've got another group of Genlisea, which I'll get into a little bit more later on. Also upper and lower lip, not so much a snapdragon form. And notice how the spur in this group points towards the back. Okay, so we've got one group of Genlisea more like Utricularia with that spur usually facing forward. And we've got another group of Genlisea with the spur normally pointing backwards, more like a Pinguicula. And lately, just to show a little bit of the information that's been coming out of this genus, we've even discovered that some Genlisea, at least one of them, forms tubers during the dry season, right? So for years, I used to think that Genlisea pygmaea was an annual. And our good friend Thomas Carroll, who's around here somewhere, went to Brazil several years ago and kept telling me that he thought he'd seen tubers. And I'm like, you're crazy, forget, no, there's no tubers in these plants, they're annuals, they die off at the end of the season. And sure enough, these are just the leaves, oops, the flowers. Sure enough, a few years ago, making some herbarium specimens, I noticed all these little tubers. Well, what a coincidence, these little tubers are sitting right under the Genlisea rosettes. I wonder what it is. And sure enough, it was Genlisea pygmaea. Although I'm going to get into a little bit later what exactly is Genlisea pygmaea. This is one of the many forms of Genlisea pygmaea, one of the largest ones actually. Probably most closely related to another species, Genlisea aurea. Anyways, this just to show a little bit of how little we know about this genus. I mean, these tubers were sitting there all the time, 200 years, and nobody noticed, except Thomas. Traps, unique traps. This genus has unique traps. Some people try to classify them more similar to pitcher plants. Oh, they've got the little lobster traps, whatever. The things go in, they can't come out. They've got the little choice hairs. It's almost like an underground Sarracenia for aquatic microorganisms. It doesn't really fit any of these descriptions. Okay, we've got the typical Y-shaped trap, okay? And sometimes some species don't have that many traps. Some of them have huge clusters of traps. And to tell you the truth, most times when you try to collect Genlisea, all the traps break off. When you try to wash them, make a nice little herbarium specimen, you usually don't get many traps. These are rare cases. This one especially was growing on a mat of algae on a lake. And this one, just painstaking washing. But notice that none of the branches are really present on this plant. Can you guys actually read the names? It looks like it's a little out of focus, maybe. Anyways, good luck. So the typical little wide traps of Genlisea. You first have a little stem that leads down to a little bulb, hollow bulb, from where a hollow tube leads down and splits into two arms, right? So there's a view of the bulbs, also called the stomach, utricule. That's where the digestion occurs of the prey, supposedly. Here's a view of the twisted arms. It's a little hard to understand what these arms actually represent. Basically, imagine just a sheet of paper folded into two, and that slit twisted around and around, and you can see the little entrances there. Look at all these minute little entrances all around where prey goes in. And inside is that hollow tube leading upwards, right? And inside that tube, what do you have? Tons of little hairs, like inside a Sarracenia. So the prey can go in one direction, but it can't come back. Now recently, there was an interesting little paper, which although not exactly about Genlisea, will help you understand a little bit more maybe what the problem is regarding understanding Genlisea traps. So in this 2006 paper, it was mentioned that Utricularia multifida, which was previously known as Polypomphalix multifida, there's 3 species in that little group, Polypomphalix. Their traps are a little different from all other Utricularia. So it's the most basal of all Utricularia, that is the most closely related in terms to Genlisea. Notice there we can see some Genlisea, oops, sorry. You can notice some Genlisea there. The most closely related taxa, most basal within all the Utricularia is that little group. So the authors hypothesized that these traps are not the typical Utricularia snap traps or suction traps that suck in prey. That's not how they work. So Utricularia multifida has nice little rosettes, very similar to the Genlisea rosettes. Here's a picture, beautiful picture in the wild showing in red the traps, in green the leaves. Alan was just pointing out to me how it's the only Utricularia that have traps facing inwards. The mouth faces inwards towards the rosette. It's a strange position comparing to all other Utricularia. And they have very thick walls. These traps and polypomphalics have very thick walls. It's not little thin walls that can easily, you know, flex in, flex out when they're capturing prey, expel all the water. These are very rigid walls. And notice over here what appears to be the trapdoor. It's not right at the entrance with a little trigger mechanism where it opens and sucks in the prey. There seems to be something else here. And that's where those authors in that previous publication were saying, well, it appears that this is actually a passive trap, not an active trap like all Utricularia. It probably acts a little bit more like Genlisea than like Utricularia. There's a little entrance towards over here where prey will, for some reason or another, be attracted in there or will go in there, force open this door, or maybe it is a little bit open, and then it's maybe like a little cat door, you know, and a dog door in the door, and a regular home door, and then they can't get out. Okay? So this sort of leads to the question, okay, if we have some basal Utricularia, which apparently don't have active traps, in fact, Andreas was telling me that, you know, he's tried poking these traps over and over, and there's no motion, there's no movement, there's no snapping suction mechanism, apparently. So how do the Genlisea traps really work? As we'll see later on, there's been some hypotheses that maybe they're active, they're sucking in water all the time, expelling it from, you know, from the traps, sort of like what Utricularia does. So the truth is that after 200 years, nearly 200 years after the discovery of Genlisea, do we know how the traps work? We don't. We still really don't know how the traps of Genlisea works. Okay? It is a very, very complex mechanism, very unique. So just to give you an idea, where are Genlisea found? At the moment, we have about 22 species accepted in this genus. Several others have been published and lumped back again, and they'll probably be divided up again, as you'll see. And they're distributed between Latin America and sub-Saharan Africa. There's about 11 species on each side of the Atlantic, but none of the species are known to occur on both sides of the Atlantic, either in Africa or in Latin America. There are perennial species, and there are annual species. Perennial species are found in typical wet habitats in savannas and up on mountains, swampy areas, boggy, margins of pools or lakes, on the border of streams, usually waterlogged areas that are wet year-round, or in the case of that strange form of pygmaea that we saw, they can actually grow in drier areas and either barely hang on or actually go dormant as tubers. Whereas the annual species tend to occur in habitats that literally dry out for part of the year, sandy or rocky cliffs, and yeah, and although a lot of these annual species, they will act as annuals in the wild, in cultivation they often become perennials. Okay, so notice over here the density of species. There's a very high density of species here in southern Africa, very high density of species in central Brazil, in southern Venezuela, and the highest density is one little place in Brazil called Itacambira, which is a tiny little spot over here where we found, if I'm not mistaken, like 7 species. Yeah, I think we have the map. Yeah, there we go, 7. 7 species in a small area. Literally, you can walk from one to the other. It's in a very small area, 7 species of Genlisea. And over here in yellow, you have what we call subgenus Tayloria. So there's 2 big subdivisions in Genlisea, what we call subgenus Genlisea and subgenus Tayloria. Subgenus Tayloria is well known for Genlisea uncinata, Genlisea violacea, lobata. They occur in a very small little area within the range of Genlisea. So just to show you a little bit of the habitats of these plants, so this is an example savanna habitat, or what we call in Brazil cerrado. Okay, very grassy, often you have some short bushes or trees, and very often you'll see these palm trees, Mauritia flexuosa, which is typical of wet habitats, and this is a great bioindicator for me. Whenever I'm driving around, I'll see these trees, like, oh, let's go over there and look for CPs. Because these are pretty much wet year-round, and you always find something growing here in the grasses below these palm trees. But you also find things, if there's sandstone mountains around, you'll find stuff growing on the cliffs at the base of these mountains. It's a beautiful place in central Brazil, which I love, Chapada dos Veadeiros. One of the most, probably the most botanized area in Brazil, the most popular place, Serra do Cipó. Amazing place, tons of species. Every time we go, we find something new. And here you can see characteristic of this habitat is it's a type of savanna, but up on highlands, sort of like what we see for tepuis, but not the big bare sandstone. It's more quartzy, sandy. You can see all of this is quartz down here. This is early morning. You can see the fog coming over the mountains. It gets very humid every night, every day. In the morning we come around, it's very dewy, the ground, although later on in the day it's going to heat up, it's going to get very dry. There's a pretty prolonged dry season. So for example, this habitat is extremely dry in the dry season. Yet every night it gets a little bit of dew and the plants take advantage of that, the Drosera, Genlisea, Utricularia. Two colleagues of mine in Brazil. Now this is an example of another type of habitat where these plants grow. On cliffs, on little inselbergs, ferrocretes, basically little isolated rocks in otherwise different vegetation. For example, the tepuis in Venezuela, northern Brazil. You got all these little isolated mountains where usually you get more of the annual species because they dry out pretty much unless they're very big mountains where you have permanent streams coming off. Okay, so this is one in Sierra Leone. And another habitat which many people don't know about, coastal habitats. We have some sandy coastal habitats in Brazil, northern South America, and I imagine this is how it grows in Cuba as well. You have all these often sand dunes. In the middle of the sand dunes, you'll have these wet habitats that are actually wet part of the year and then will dry out. It's very dark, peaty water often, similar to what we find up on tepuis or other sandstone highlands. And there's a lot of CPs in these areas. Not just a lot of Utricularia, some Drosera, and even we've discovered recently some Genlisea. So what's up with that big subdivision that I was telling you guys about? Yeah, there's two big groups in Genlisea. One of them, subgenus Tayloria, opens its seed capsules, like some Utricularia do, like Pinguicula, it just opens in half. Whereas the other subgenus has a unique way of opening its capsule. It opens up like if it was a globe at the equator, and maybe more longitudinally, yeah, a little bit more latitude further up. Although there are reports of spiral openings for some species. Okay, but so this is the big characteristic that divides the two subgenera. And at the beginning, when you see these things, oh, how stupid can that be? You know, does that mean anything at all taxonomically? You'll see afterwards that this is based on the phylogenetic work that's been done with DNA sequencing. It is a very important subdivision within Genlisea, not just taxonomic bullcrap. Anyway, so subgenus Tayloria. It was originally published as section Tayloria with only one species, Genlisea violacea. Genlisea violacea was one of the first Genlisea to be published. We only got two more species in this group much later on, 1983, 1989, Genlisea uncinata, Genlisea lobata. They're perennials or annuals, have glandular ovaries and a few other characteristics I'm not going to go into too much. The flowers salveriform, like I was saying, they're not exactly snapdragons, close to it, but Genlisea violacea very popular in cultivation. There's many different forms. I've come to the point where I can almost recognize where the plant is from, which mountain, based on the flower color. There are literally a lot of different forms of Genlisea violacea. Genlisea uncinata, probably one of the biggest species in the genus, has huge flower scapes. In the wild, I've seen them up to 80 centimeters tall. In cultivation, I've seen them up to 1 meter 20 in length. They got pretty large flowers with hooked spurs, grow in very remote places in northeastern Brazil, up on mountains in very dry areas, but up on those mountains they get fog every morning, no matter whether it's, you know, really in the middle of the dry season, they'll still get a lot of fog. And Genlisea lobata, very tiny little species, very similar to Genlisea violacea in ways. Okay, very similar habitats, usually grows more on granitic outcrops, whereas Genlisea violacea is more on the sandstone highlands. But in this group, we've been finding a lot of variation, let's say, not just the Genlisea violacea, but a lot of other little plants which don't fit any categories. We've given them some little informal names. Hopefully we'll publish them soon. But anyways, we've got one little tiny thing known only from two populations, probably more widespread up on those same mountains where Genlisea uncinata grows. It's got a strange little fat spur. The flowers aren't even totally closed. They're sort of semi-open always. We have another thing, the giant violacea, pretty common in cultivation. It is like a larger form of Violacea, but at the same time very different. It's got some very unique characteristics, more or less geographically separated as well. Recently we found another one, pretty large species, only known from single location at the moment, from that place called Itacambira I mentioned, where 7 species grow together in a relatively small area. Beautiful plants. There's a tiny little thing which I only know from a single waterfall at the moment. Sorry for the bad pictures there, very old. Tiny little thing, only grows on a few rocks at the base of one waterfall. That's the only place I know where it grows. Tiny, tiny, tiny little plant. Smaller than lobata, smaller than Violacea. And we've got another large species similar to Ensenada, except it doesn't have that hooked spur. It's geographically hundreds of kilometers away, only known from 2 locations actually. And there's all those different forms of Violacea that, you know, what do you do with them all? Is it just one big variable species, or is it a whole bunch of different species? How do we split them up if we want to or not? Should we keep them all together? So here's a little, here's the phylogeny that was published recently. Okay, let me see. Here we go. So interestingly, that subgenus Tayloria that we were just looking at, Violacea and Ensenada, those guys, and some of those undescribed species here, they're way at the base of the Genlisea family tree, closer to Utricularia and Pinguicula. Okay? And then we have a group of more derived species, which we're going to be delving into a little bit more now. Okay, so just here a closer view. Notice that we have Genlisea Ensenada as the most basal. Then we have that giant Violacea, that Itacambira plant. And then we have a whole bunch of different Violaceas. These numbers are just different collection numbers. Don't worry about them. And notice, lobata. It's sort of in the middle of the Violacea. So again, is Violacea a single species? This seems to say no. We might have more than one species there. And like Alan was saying before, I completely understand that little drama there. I mean, there's been several names published for the Violacea group. And, you know, do any of them match any of these forms? Taxonomically a little complicated. So lobata, it's right there in the middle of the Violaceas. What do? Do we call it all Violacea? Or do we split those up into several different species, with lobata and Violacea being 2 of them? So recently with this phylogeny, we noticed that there were a few natural groups. Yeah? So 3 new sections were published. So all the remainder of the Genlisea were split into 3 sections. Section Genlisea, section Recurvatae, and section Africanae. Where do these plants occur? These different groups. So again, Tayloria is only found in southeastern Brazil. And then we have the African species here, African species up here, right? Now notice that there's a few different branches in there, and the most apical branches are actually Brazilian again. So wait a second. So the most, let's say, primitive, the most basal of Genlisea are in South America. Then the slightly more derived ones are in Africa. And then the most derived of all are back in South America. So wait, what's going on here? So usually the most basal suggests possibly, sometimes, where the point of origin was closest to. Right? So this would sort of suggest, okay, maybe Genlisea had its origin in South America, maybe even Brazil. But then we have a derived group that is present in Africa. So what happened? A long distance dispersal, or was this even before the continents broke up? You know, and they were just growing side by side at that time. Although it doesn't seem like Genlisea is that old. Okay, so it seems like it's most probable that there was some sort of, I don't know, long distance dispersal. Little seeds up on a storm, birds, who knows? And this originated 2 of the sections, section Africanae and Recurvatae. But then within section Genlisea, some of the species seem to have gone back, or at least one plant migrated back to South America and originated all these other species. So it seems to have skipped, possibly, across the Atlantic at least twice. So what do we have what are those 3 little sections? So one of them is, like the name says, it's African. Got several species there with their dates of publication. Some of them very recent. Notice that Bartholatiae, Tayloria are very recent species. And there's the boring little taxonomic characters that define this group. Okay, some of the species were not in the phylogeny because, of course, they grow in such remote places that we haven't really been able to get material for these plants yet to do any sort of DNA sequencing. So in this group, if you look at these pictures, you think that it's all the same species probably, right? Well, no, these are all the different species. Okay, only one of them looks terribly different than the others, but yeah, for us taxonomists, when we look, yeah, the spur is longer in one is hairier here than there, whatever. These are pretty good species. You see over here a Drosera elongata growing with this Genlisea subglabra. Okay, so these are the African species that we have pictures of, that we know what they look like in the habitats from Section Africanae. And these are the two that we don't really know what they look like in habitats. We only have herbarium specimens from the early 1900s, Genlisea taylorii and Genlisea angolensis. So these are not in cultivation yet as far as we know. Then we have that other little section, recurvate, where we have Genlisea glandulosissima, margareta, and pallida. Pallida, same thing. We don't really know what the plants look like in the wild, very remote places where it grows. Only known from a few collections in Angola. Now, there was a little pallida going around cultivation a while ago, a so-called pallida. If you think you have pallida, sorry, you don't. Okay, pallida, as far as we know, is not in cultivation. The plant that was going around this pallida is actually Genlisea aurea. So the other two species, glandulosissima and margarete. I was very happy to be on a trip a few years ago with Andreas Fleischmann where we rediscovered these species in Zambia and saw how they were growing in the wild. Amazing little species. Glandulosissima usually forms tufts of flowerscapes. It's compact groups of flowerscapes coming out of the rosettes. Very glandular plant, like the name says. Margareta is known from a wider range that occurs in Madagascar as well. It's been in cultivation, I think, a little while longer, maybe. Now, the interesting thing is that when we sequence these guys, Genlisea margareta seems to, one of the collections is clustering more of the Genlisea glandulosissima. They're one a little bit further apart. Are we dealing really with one species, two species? Actually, what seems to be happening, Genlisea glandulosissima and margarete apparently do cross sometimes in the wild. Okay, this would be probably the first case of natural hybrids between Genlisea, although one other may exist. There is one possible record of it, angolensis with pallida. Okay, so what we're seeing here might be the result of hybridization between these two species. That collection is from an area where both species grow, whereas the basal one here is the one from Madagascar, so a little bit more separated from glandulosissima, maybe a little pure genetically, not as much mixture. Now, if you thought all those African ones, purplish ones, look the same, wait till you see these guys. So Aurea, Filiformis, Pygmaea, Repens, Genensis, Roymensis, and a lot of these are the yellow ones that you guys are familiar with from South America. Some of them a little bit different. So we have, if you look over here again, Genlisea guianensis, Sanarapuana are the basal ones in this group. Maybe not a coincidence that they actually look more like the African species, like Margarete, glandulosissima, have the purplish flowers. Now these two both have usually very long leaves, 10 centimeters, 15 centimeters, reddish leaves right on the surface sometimes, these huge traps right on the surface as well, very large traps. Notice that although the flowers are purplish, this is the guianensis that grows south of the Amazon, and this is the guianensis that grows north of the Amazon. And these did split rather nicely over here in our phylogeny, so it seems like we may be dealing with two different species there. Very widely separated geographically. Genlisea aurea, one of the most popular Genlisea in cultivation. Here's a nice little population near my hometown, São Paulo. And unfortunately, our friend Studeniczka did not come. This is him and a few of his Czech friends with me at this beautiful Genlisea aurea population where we had tons of flowering scapes. Notice that it's very foggy, very rainy. We've all got our umbrellas out. Here's a picture from this location. Notice how many flowers are on this flowerscape. Genlisea aurea is famous for having very compact rosettes covered in slime. There's this mucilage on the leaves that we don't really know what it's for. Is it there to protect from predators, whatever? It's just, we don't really know what that mucilage is for, but it's very thick sometimes. Most of the times it's very thick on the rosettes. But other times we find rosettes that are pretty dry, no mucilage. And slightly different leaf shapes. I don't know if you can see this, a thin little strap shape. This is more, I don't know, spoon-shaped or, yeah. Is it the same thing? Sometimes we see them growing underwater, sometimes in drier, sandier places. I've seen them up to almost 3,000 meters altitude and down to about 500 meters altitude in really hot areas. I remember one place where, man, it must have been 45 degrees Celsius. The water was coming down the seep. It was heating up so much with the sun that literally if you stand at the bottom of the hill, the water was hot. It was hard to stand in, and yet it was covered with Genlisea aurea, all at the bottom, Drosera communis, Utricularia, and this is very hot water. And often we'll find rosettes in the wild that have a lot of leaves floating at the surface. I never really knew what to think of this. You know, is that some animal stepped on it and all these leaves came off, or is this natural? Andreas says he actually saw little plants growing on these leaves in the wild. So is this maybe a way that the plant reproduces naturally, vegetatively? Possibly. And the flowers. We have a lot of differences in the flowers, although they're all yellow, I know. Notice you don't see a spur on this guy. It's hidden behind the lower lip. And yet on other forms, you see huge spurs. And some of them, they're just slightly poking out. And notice what a fat lower lip this guy has, with a narrow lower lip. Again, we might be dealing with different things. In the phylogeny picture that we were seeing before, notice that the 4 collections of Genlisea aurea are pretty well separated here. So again, we might be dealing with more than one species. And I'll show you later, there's a lot more differences between these guys. Very surprising little group. Okay, so a lot of variation in there. Genlisea glabra, ooh, this is an interesting one. Best of all, because it's not yellow. So this is one that was only known from a single mountain, Chimanta Tepuy in Venezuela. And Andreas was there recently and found that was the habitat shot. Along these little waterways, you have the little rosettes. And it's a curious little plant. Although all the other ones in this group are yellow, these are sort of pinkish, lilac-ish, whitish, whatever. It's got very dark flower scapes, oops, with these little pendulous tops. Very unique little plant, very different from all the other ones in the group. Then we have Rora immensis. Grows on the top of Tepuys, very high altitude plants, known from a few little mountains there, Venezuela, Brazil, Guyana border. It's almost like a small little Genlisea aurea, actually. Beautiful little plants. Repens and filiformis, very widespread plants, probably the most widespread species in Latin America. Okay, so repens, interestingly, sometimes forms a rosette and often forms a trailing stem. I don't know if you can see here how the leaves are spread out along the ground. Has a trailing little stem, like your Utricularia. And here you can see the leaves spread out along the ground, the flower scape coming up. Here's a flower. You can actually see where the fruit is going to open up, like I said, at the equator and 1 latitude further up. And I don't know if you noticed in that phylogeny picture, which I'm not going to go back to at the moment, we sequenced 2 different Genlisea repens, one from north of the Amazon, one from south of the Amazon, and they showed up in 2 different branches on the tree, one closer to Pygmaea, if I remember well, one closer to repens. So again, apparently we're dealing with more than one species here. Is it maybe that the rosetted ones are one species and the long-stemmed ones are another species? And we have filiformis. Luckily, this one is not too variable. This is one of the few species where probably it's pretty easy to identify. It's got these fat little spurs, okay, huge spurs, sometimes a little purplish, obtuse tip. This is an annual species, whereas repens perennial. This one grows all the way from southeastern Brazil all the way up to southern Mexico, going into Cuba as well. Pygmaea, okay, this has been one of the headaches. So whereas filiformis is probably one of the most stable species, not much variation. Pygmaea has been a huge problem because we found some Pygmaea-ish things growing on the coast of Brazil. Little pointy spurs, little small flowers. We've also found, where is it? The tuberous Pygmaea, huge plants with mucilage on the leaves, similar to Genlisea aurea, huge spurs, tubers. Look at this, a picture of the seedling that Andreas took in cultivation. I don't know if you can see the seed right over here. It puts up a few leaves, puts up a trap, down a trap, and it's already forming a tuber. It's barely germinated, it's already forming a little tuber. And here's another form of Pygmaea, which I usually call the medium Pygmaea, because I always used to call this guy the large Pygmaea, this is the medium Pygmaea. Unfortunately, I do not have pictures of the tiny Pygmaea, the smallest one of all, which actually seems to be the type Pygmaea, the true Pygmaea. Unfortunately, this one I do not have pictures to show you guys, but it looks probably more similar to this guy over here. It's a small little plant, but even tinier. It literally is, I think, the tiniest Genlisea in the whole genus, even smaller than filiformis. Usually grows like 2-3 centimeters high, the whole flower scape, a tiny little yellow flower. Not a very easy plant to find. It's pretty widespread, but not very easy to find it. You don't find large populations. You find one here, one there. Tiny little thing. And why is it so difficult to identify these plants and find out which is the true Pygmaea? Because again, the type herbarium was collected nearly 200 years ago. It's been in Europe while we've been investigating these plants in Brazil, and the original plants are sort of smushed and cracked, and you only have a flower scape and not the leaves or bits and pieces of the plants. So, here's a little graph. I want to show you Genlisea publications over the last nearly 200 years since the genus was described. So the genus was originally described in 1833 by French botanist Saint Hilaire after his visits to Brazil in the early part of that century. He not only discovered the genus, but he described the first few species, Aurea filiformis minor, Pygmaea violacea. Minor became a synonym of Aurea afterwards. We might have to split it again. So he published the whole genus with a few species from Brazil. And as you can see, number of publications here versus a year, not much happened with Genlisea in the first nearly 100 years after its discovery. Nobody seemed to be really interested in this genus. Most of the stuff was just, publications of species. Yeah, this species, that species, a lot of them became synonyms. Oops. Okay, not much happening there. Although we did have some important work. Warming, Darwin, Goebel were the first to really start looking at these traps and thinking, wait a second, what's going on here? Could this possibly be carnivorous? Could this possibly be trapping animals? Although Darwin, Warming only looked at herbarium specimens. They didn't have live plants. So Goebel was the first one to really be able to look at live plants and understand these traps a little bit better. Now, from 1900 to 1971, yeah, it picked up a little bit. We got a little bit more publications going on. One year, we even had 3 publications. When was it? 1957. But most years, either no publications or 1 publication. Again, not much going on. And most of these publications, new species, taxonomic revisions, the flora of Guatemala, flora of this place or that place, that's basically all there was. That is, most of this work is just looking at herbarium specimens. Okay, so you have these botanical expeditions, people collect everything that's out in the wild, and then afterwards these guys come around like, oh, what are the Genlisea of Honduras? There's no real work in the field here. Okay, people are doing botanical expeditions, but nobody's really going out in the field and studying Genlisea, it seems. With a few exceptions. Okay, 1939, 1942, important works here. Everybody knows Lloyd. They started discussing what sorts of prey are inside the traps, did a little bit more detailed morphological analysis, hypothesizing how did the animals get in there, what exactly is inside the traps. Finally, starting in 1972, things picked up. Look what happened. All of a sudden, we start getting 2-3 publications a year. And in the year 2000, we started getting, look at that, 4, 5, 6, 7 publications a year. Okay, this is where I was joking that things came out of the closet. What's going on here? All of a sudden, the last 10, 20 years, publications of Genlisea have boomed. Although in the 70s and 80s, it was still mostly species publications. Revisions, we have from Trinta from Brazil doing a lot of important work, but still mostly just looking at herbarium specimens. Okay, we have a lot more flora descriptions and other stuff, and this has been going on, like you saw, all the way from the discovery of Genlisea, but notice what's going to start happening now. We're going to start seeing some papers on ecology. Only more towards the end of the 20th century, we start seeing papers describing, literally, okay, how does this Genlisea grow? How does that Genlisea grow? What are those plants are going with it? And what sorts of habitats are they, and not just reading the herbarium labels, saying, oh, this plant grew in a savanna, whatever, a kilometer of whatever road. These are actually works where people are going to the field and looking at these plants in the field. I'll never forget, in my early days in carnivorous plant hunting, my first few times out in the field in the early 90s, and reading like, Peter D'Amato's book or other books like that, saying, oh, Genlisea, extremely rare. And I'd be like trampling all over populations of Genlisea violacea, Genlisea aurea, huge populations everywhere. It's just, nobody had really bothered to go out to these places and study these plants in the wild. We're just doing, almost all Genlisea work was based on herbarium specimens. So, all of a sudden, we get a boom as well in studies in morphology and trapping mechanism. Why? Because we started having more access to live plants, studying things that we couldn't study in herbarium specimens. We start getting discovery of digestive enzymes. Look at that, 1975. We're beginning to get closer to a final proof that maybe this plant is carnivorous, Juniper, Robbins, and Joel, very important work. Detailed morphology of the traps. Reut as well. Barry Myers-Rice in 1994 started speculating as well about, is this an active trap? Is it a passive trap? Do animals just go in randomly? Or is the prey literally sucking up water, creating a constant flow of water inside the traps to help draw in prey and to help keep the nutrients from digestion within the trap? Because remember, there's that little hollow vesicle and a tube leading all the way down to the entrances where the prey goes in. So wait a second, if it's digesting prey up here, aren't the nutrients just going to dissolve down and they're going to be lost? So is there maybe some sort of water draw into the trap to keep these nutrients from diluting out and to help draw in prey maybe? And then other important work, look, we got Studnicka saying that maybe there's two types of traps, some that stay closer to the surface, capturing one sort of prey, and other traps going deeper into the soil, capturing maybe other sorts of prey. Why would it need two types of traps? And then we have an important work here in 98, where finally somebody showed, okay, this is capturing active, it's trapping something. We put, protozoa in the water, and they were literally going into traps. Whether they're attracted to it or not, why they were going inside, we're not really sure. But they're actually going into the traps and apparently being digested and being absorbed into the plant. So hey, final proof of carnivory, it seems. 1998. When are the plants discovered? 1833. Took a while, huh? So notice, one of these, 1975, but most of them getting into the 90s, late 90s. A few more papers on morphology and trapping mechanism. Again, Studnicka. Maybe the traps just form little holes in the compact sand or gravel where they grow, and it's just what naturally attracts the prey inside is not some sort of chemical which smells good to these creatures, but it's just little holes that they want to swim in. One of his hypotheses was maybe, underground, not much oxygen, and this creates an oxygen-rich environment which attracts prey into the traps. And then a little bit more discussion, Adamek, is there water flow inside the trap or is there not? Although the studies were usually done with traps detached from the plants, a lot of these studies, so whether that's really valid or not, we'll see. Glandular hairs, what sorts of glands do we have inside the traps? Are they producing enzymes? Are they producing mucilage? What sort of things are we seeing there? Feeding behavior in the lab, what sorts of things are they eating? How did the prey die inside? Look at that, 2007, we have a paper by Adamek there. We're suggesting, okay, maybe prey is killed just by lack of oxygen that little stomach pouch in the trap. Maybe they just suffocate in there, lack of oxygen, it seems. And more about hairs, attraction. Oh, interesting one, 2008. There we go, algae commensal communities in Genlisea traps. We're going to get back to this later on, but both Utricularia and Genlisea seem to have commensal organisms within the traps. We'll get back to that one in a while. And again, functional anatomy of ovules. Okay, that doesn't sound too interesting, but notice that all of these papers, all these publications, without live plants are nearly impossible. You need live plants to study this stuff. All of these were only possible, again, because only recently we started getting these plants into cultivation. People had these available in their labs to pull them apart and do all these tests on them. Otherwise, with smushed herbarium specimens where the traps are usually not even present, you can't really do much. Okay, so these, in the latter parts of the last decade, in the last decade, this is one really cool test that I liked in CPN several years back, Studniczka, where he cut the traps of Genlisea. Again, it's detached from the plant. Maybe it works a little bit different when it's attached to the plant, but he cut some traps over here at this point and some of them right through the little stomach. And he dipped them in dye. So as a control, he put a little capillary, and you can see the dye going up into the capillary. Now the traps that were cut at this point, notice that they do suck up the dye. Interestingly, the traps cut over here do not suck up the dye. So apparently there's some little glands in this area which produce some sort of mucilage that keeps the dye from diffusing out of that little pouch. So why did he do this? Because he wanted to try to understand the products of digestion. It's digesting prey, but yeah, again, is it diffusing out of the trap? Apparently not. There's something holding it back. There's maybe a little mucilage barrier which prey that is swimming actively can go past, but once it's dead and being digested, its products can't diffuse out. Notice how complex this is becoming now. Okay, we had what was a passive little trap, all of a sudden becoming something that might be at least semi-active, possibly drawing in liquid. It's got also maybe a little mucilage barrier in there to allow prey in, but not allow the digestion products out. It's possibly pumping water out within from that vesicle, who knows? So back to the scheme. We have tons of publications in the last 10, 20 years. Of course, having a lot of live plants makes it easier to extract DNA and do phylogenies. So all of a sudden we start seeing the first phylogenies done with Genlisea. Okay, initially with 1 or 2 species, it grew, more and more species were added until the latest one that just came out this year where we got almost every single species of Genlisea, except the 3 which are isolated out in Angola, which we don't have live material of, unfortunately. Few herbarium specimens, and it's not always easy to get DNA from herbarium specimens. But one of the most important papers ever published, I would say maybe even with carnivorous plants, came out in 2006. Probably by accident, these guys discovered in 2006 that Genlisea has one of the smallest, if not the smallest, genome among all flowering plants, all angiosperms. Okay, it's a tiny little amount of DNA per cell. Tiny, tiny, tiny. And some of those chromosomes are so small that they're the size of bacterial chromosomes. So what does that mean? Why is that so important? Well, let's compare. Let's look at yeast. Yeast has a total genome size of about 12 million base pairs, 12 million A's, T's, and G's connected in chromosomes. Arabidopsis. Arabidopsis is a model plant. It's used for tons of botanical studies because it's a simple little plant. It's got a very small genome. People used to think it had the smallest angiosperm genome, 157 megabases, 157 million A's, T's, and G's per nucleus per cell. Now, people, like this little geeky guy over here, we have, as you may remember from the Human Genome Project about 10 years ago, 3.2 billion A's, T's, and G's in our chromosomes per nucleus per cell. 3.2 billion bases, which are our blueprint of life. To create another human being, you need those 3.2 billion, although this one probably has some very significant deletions. So a complex organism, now how much DNA do we need, and what is in all that DNA? A lot of DNA is repetitive DNA. You've probably heard this, that most DNA that we have is junk, only 1, 2, 3% are genes. And for our genome, that's pretty much the case. Yeah, only a small percentage is actually genes that will code proteins, whereas all the rest is apparently junk. Apparently, okay? The truth is we don't really know what all that other DNA sequence is, but at least it doesn't seem to be coding genes, maybe regulating the genes. But anyways, it does seem to have some importance because a lot of organisms have tons of DNA per cell, and it's costly for the cells. Okay, so if yeast has 12 million bases, Arabidopsis 157, we have 3.2 billion. You think, oh, we humans, we're more complex, we need more DNA, right? Well, look at that. Angiosperms, flowering plants, in general, the average is about 6 billion bases, about double the amount of DNA that it takes to make a human being is the average for angiosperm, and some of them just go ballistics. This Fritillaria, 124 billion bases per nucleus per cell. Why the heck does it need so much DNA? And where did Genlisea fit in all of this? So this paper from 2006, it measured the genome size of Genlisea aurea and margaretae, and it found that Genlisea aurea had only 64 million bases, and margaretae 63 million bases. That is a tiny amount of DNA to code for a whole flowering plant, especially if you think how complex it is with the trapping mechanism, enzymes, whatever. How can it get by on so little DNA, and why does it have so little DNA? And if you compare Genlisea with Fritillaria, you see there's a 2,000-fold difference in genome size between these plants. What the heck is going on here? Okay, and just so you know, the plant used in that study may actually be Genlisea aurea, so the genome size that was measured may actually both be for Genlisea aurea. And are all Genlisea the same? Apparently not. A few other Genlisea aurea populations are being looked into, and there seems to be a huge variation. One of them was measured at 120 megabases. That is, this one over here is 63. There's another population that has double the amount of DNA. Another one had 90 megabases. And then there's this one, which is still not published. Hopefully our colleague Todd Michael will publish it soon. Apparently 46 megabases, a new world record. Again, how can we have not only such small genomes creating such a complex organism, but how can there be so much variation between populations of what supposedly is the same species? And again, where do other Genlisea fit in? Well, other species of Genlisea are much bigger. 1.5 billion, 1.6 billion. That's a huge variation from to 1,500. Pinguicula, sort of intermediate there within that range of Genlisea, at least the species that we've looked at so far. Okay, 487 to 829 million bases. Utricularia, a little bit smaller than Pinguicula, and also some very tiny genomes. Utricularia gibba is the smallest one we found so far, and the same thing's happening with that species. It seems like every population has a different number. So, but wait, don't they all have the same genes? The plants all look the same? All the Utricularia the same? Maybe the genes are, but what's in between the genes? Who knows? And this has created such a buzz in the scientific community, outside the carnivorous plant scientific community. I mean, it's a huge thing to discover that a plant has such a small genome. It's created a huge buzz coming out in Science magazine. A review talking about, okay, what do genomes really do? What does all this DNA really do? Why can some plants get by on such small, tiny amounts of DNA? Why do some need a huge amount of DNA? So here's this article talking about Genlisea aurea, how it has a tiny little genome. And the most important thing about this paper is that this is the closest I'll ever come to publishing anything in Science. I got a picture in it. Okay, so there's a huge buzzword out there. Genlisea aurea is in the mouth of so many scientists. I've bumped into people, you know, different scientific backgrounds, whatever. All of a sudden they tell me that they want to sequence Genlisea aurea out of the blue without me even mentioning carnivorous plants. I'm like, you got the person. Genlisea aurea is not as widespread in cultivation. I mean, a few of us have it, yeah, whatever, but all of a sudden we've got all these scientists calling, asking, oh, we want Genlisea aurea, we want to do studies on genome expression, evolution, whatever. We need some material, Genlisea aurea, utricularia. There's a huge buzzword out there, and we're definitely going to be seeing tons of publications coming out in the next few years. So Genlisea aurea, oh, a little bit more. No, I'll get back to this. Genlisea aurea and maybe other Genlisea utricularia have the potential of becoming a very important model organism for science, for the understanding of flowering plants. You know, when we want to play around with corn, make a new GMO corn, or, you know, understand how to make better crops, often they'll go to model organisms, Arabidopsis. And we have something that's even smaller, has less junk in that DNA, it makes it easier to study. So with this discovery, Genlisea aurea and other Genlisea and utricularia may become very important model organisms in science, understanding how plants work, crop plants and other plants. Another very important paper, which sort of is a direct result of that genome size paper. So 2006, Genlisea aurea, Genlisea margarita, smallest known genomes in angiosperms. Already in 2010, we're getting this paper from scientists who first heard of Genlisea ever in their lives from that 2006 paper. So they extracted total DNA from Genlisea aurea, and until recently, it was only viable to really sequence small bits and pieces of DNA, 2 genes here, 3 genes there. I mean, the Human Genome Project, I don't know if you guys remember, 10 years ago, was a huge undertaking. It was dozens of labs, dozens of people, billions of dollars to sequence one human genome. Well, in the past 10 years, the prices of DNA sequencing have plummeted. We've got a whole new generation, a whole new technology, several technologies actually, sequencing DNA at a scale that we've never seen before, very cheaply. You can do single human genomes in a few days in a single lab by a few people. And not the huge, huge groups that were necessary before, for example, when we did the Human Genome Project. So this is one lab in the US. Actually, I worked with these guys. They're a customer of mine. They needed Genlisea aurea. I helped them get it. They extracted DNA from the leaves and the traps. Here's an important detail, and the traps. You'll see why. And they used one of these second-generation DNA sequencing instruments to sequence that total DNA. Okay, let's see what's in there. Let's sequence the whole genome. You can do that now. Whoops. Uh-oh. So curiously, by extracting DNA from the whole plants, including the traps, they not only obtained DNA sequences for Genlisea, but they came up with some other DNA sequences, which normally you would think are contaminants. But it's a little bit of a coincidence. All these contaminants were nitrogen-fixing bacteria. And as I mentioned in the beginning of the presentation, there was a publication done with utricularia showing that there are commensal organisms living within the traps of utricularia. And that also carbon is secreted into the traps of utricularia. So apparently the plant is feeding carbon to these commensals in the traps, and that they serve some function in the trap, maybe helping them digest the prey in there. Who knows? So apparently this is also present in Genlisea. So not only utricularia, but Genlisea have little tiny microorganisms living within the traps, helping to fix nitrogen, and they're being fed carbon from the photosynthesis of the plants. They're being fed this carbon that's secreted within the traps. There seems to be a very complex little food web going on in there. Okay, so what was simply a passive trap, notice how far it's moved along, you know, to a possibly active trap, to a possibly little chamber with an active food web going on in there, where there's commensals living in there and helping digest the prey maybe, and being fed directly carbon by the plants. So within that whole genome, Genlisea aurea, about 64 million bases, amazingly, genes are about half that amount. Now remember the human genome, about 2–3% are genes. Whereas in this piece of it, about half the DNA is genes. So all those repetitive sequences or other garbage DNA, which we don't really know what they're for, are missing. And it has about 20,000 to 30,000 genes, which is not very far from us. We're also around 30,000 genes, if I'm not mistaken. So you need about the same number of genes to make a human as you do a flowering plant. Yet the total DNA per cell is very small in these plants. So Genlisea aurea does not seem to have fewer genes than other plants. It didn't lose genes in this genome reduction. And it's basically just genes and not much repetitive DNA in there. What else? So why such small genomes? What's going on with Genlisea utricularia? Why do they have such reduced genomes? How can they get by with such little DNA when almost every other single species on the planet needs tons of DNA with a lot of so-called garbage DNA in there? Okay, Genlisea and utricularia, I don't know if you remember, have very small genomes. Pinguicula a little bit bigger, some Genlisea a little bit bigger as well. So why are Genlisea utricularia so different from Pinguicula and even extremely different from all other flowering plants? I was trying not to get too technical here, so let's see if I did a good job or not. But there is a paper saying that there's this one enzyme called cytochrome C oxidase in utricularia and Genlisea. It has a rare mutation. Only in utricularia and Genlisea. Looking through this huge database of this gene that was sequenced for tons of different organisms, only one other organism, I think, was found that had the same mutation. And it seems to be a mutation that elevates the energy production at certain periods within the traps. It increases respiration in detriment of photosynthesis, it seems. But for some reason, it's important to increase respiration at certain moments. I'm assuming maybe immediately after prey capture, when it's pumping out water from the traps, whatever, in utricularia. Oops. So this increased amount of respiration in the traps of Genlisea and utricularia also, as a side effect, generates highly reactive oxygen. Highly reactive oxygen is bad. It's terrible. Free radicals. This breaks up DNA. It's extremely damaging to DNA. So this is a little side effect. Apparently it was very important for the traps to be able to get all this energy all of a sudden, but it seems to have a very bad side effect. It's damaging. It's like radiation. Okay? It will cause base changes. It will cause mutations in genes, which explains what actually Andreas had so much trouble with sequencing some of these Genlisea, where the sequences were so different from even within the same species. You had to keep repeating it and changing the DNA sequences that we call primers to amplify that DNA. There's a really fast evolutionary rate in utricularia and Genlisea, at least for those genes that we were studying, more than any other species. They're evolving extremely fast. Part of this seems to be because of all this damage that's being caused to the DNA as a side effect of this enzyme, of that little mutation we have in that enzyme. So as a side effect, it's creating all these mutations, but somehow the plants are able to repair it and stay alive and not go extinct. So they have this negative side effect apparently, that their DNA is being damaged all the time, but they must have a very good fixing mechanism where they go in and fix it up. But sometimes some of these mutations are fixed if they're not really bad. But there's another side effect where it not only causes mutations, it literally breaks chromosomes. It breaks them. So where you had 2 pairs of chromosomes, you might all of a sudden have 8 chromosomes. It makes tiny little breaks, which are sometimes repaired, but oftentimes not. One thing that's not published yet, but what we're seeing is that the plants with the smallest genomes are the ones that have the smallest chromosomes, the larger number of chromosomes. So another negative side effect, apparently, is that all this reactive oxygen is breaking up the chromosomes, and in this breakup, it loses a lot of bits and pieces. Of course, if it's a very important bit and piece of DNA that's lost, the plant's going to die. But apparently it can get away with losing a lot of DNA and becoming a very, very tiny little genome without actually dying, actually surviving somehow with only 64, 63 million bases, even 46 as you saw. Okay, so the constant mutations, the constant breaking of chromosomes followed by imperfect repairing apparently is causing a reduction in the total amount of DNA by deletions, little bits and pieces that are lost, and also causing a large number of chromosomes in these species with the tiny genomes. So tiny genomes, large number of chromosomes, often very tiny little bacterial-sized chromosomes. What exactly is going on there? Hopefully we'll have more published soon on chromosome numbers in these Genlisea. Very difficult to count because they are so small. But yeah, you can be sure that there's going to be a lot of interesting information coming out from Genlisea. And that, as I said, this is a buzzword in the scientific community at the moment. A lot of people are researching Genlisea utricularia and are going to be doing a lot of interesting studies. that will be a reference for a lot of other studies with plants, non-CPs, in the next few years. So a huge thank you. This is the end of my presentation. Just a huge thank you, everyone, to the Hortus Botanical Garden, Carnivora, and ICPS. Really appreciate all the work you guys have put into this conference. My friends who've not only helped me in the field, but have helped with pictures. Here's one of them, Vitor, with a beautiful population of Genlisea guianensis growing at the edge of a little pond, the traps flowing into the pond. And of course, Andreas and my other friend Todd Michael, who helped me with slides for this presentation. And thank you all for staying awake for this last presentation. Questions? Anybody, any questions? Hope I didn't get too technical on you in the end. Nobody?
Just one quick. The tuberous form, yeah, it just looked like basically it was growing in the area that the stomach, whatever you want to call it, would be. Is that what it is?
No, it just looks from the photograph. Took the one I sent with the worst angle. It's in a separate shoot.
Let me see if I can find it.
The dress behind.
Yeah, no, it's actually a little stem, and another picture I have, you can sort of see it comes out from like the center of the rosette, turns downwards, and forms out of the tuber.
It's all in the angle.
Very interesting when it germinates. The first thing it puts up is a little tiny trap leaf which doesn't grow far, just to anchor it. Then it produces the first photosynthetic leaves, then it produces the first trap leaf. Then the next photosynthetic leaves, but already this tuber. So you could already, after several weeks of growing, you could already survive dormancy.
Oh, here it is. Now let me open this up. Let's see. Oh, not this one. Sorry. Yeah, but it's much larger than that little pouch for the traps. Much larger.
Yeah.
That little pouch is like a few millimeters wide sometimes.
And solid, like a tuberous thruster tuber.
Yeah, no, like you said, it's all in the angle. That's all right.
I think I have it here. Genlisea aurea. It's the next one. Yep. Here we go. This is it. So let's look at that picture again. Am I doing on time? Okay. Yeah, it actually is. You're right. It looks like it's right over where that pouch would be, but you can see a separate little stem here, and this is the trap coming behind it.
Yeah.
You see the chamber right up there. It's a little more dark.
Is it? Oh, okay, so the pouch is not here, it's up there.
Nope.
Anyways. It's a tuber. See if I forgot anything. Yeah. Any other pictures? Question?
The reactive oxygen that's being formed by the mutated enzyme, does that play a role in the digestion, or?
As far as I can tell, as far as from the papers, it's important for generating a large amount of energy at specific moments. So respiration, increased respiration rates at certain times. So like it can trigger very quick respiration rates. That's the importance of that mutation apparently. And why would that be important? I don't know. I imagine something with digestion of the prey when it captures prey, like in Utricularia.
In Utricularia, it's especially important to make the trap active again.
Yeah.
To pump out the water. That's when this.
Okay. So that would suggest that it is an active trap.
I don't know. Yeah, it's a highly reactive oxygen.
When you get highly reactive oxygen, it also goes to the non-reactive one called ozone.
Yeah, that's
Yeah.
yeah, something like that. Basically, it's breaking up the DNA. Yeah. It's basically apparently what's causing all that, reduction in size of the DNA. Oh, yeah. Anybody else? Questions?
When you were talking about the two Atlantic crossings, do you have any sense of, or I assume because the genomes are evolving so quickly, we don't have a very good sense of the molecular clock data on when they merge?
I think Andreas can answer you that one.
Yes, we have big problems because they are linealer, so a very highly evolved plant group, which is evolving very, very fast. We only have 2 fossils for the whole plant group of, well, snapdragon-flowered plants. And interesting, one of these is Biblis. There's a fossil Biblis seed. Biblis belongs also to this group.
There was, because they lost the seed. Remember, it's in the paper. You have the drawing of the seed, but somebody dropped it on the carpet and never found it again.
No, he actually had a photograph.
And the other one is the tree, which belongs there. But as the group, the Genlisea, is so derived, we cannot align sequences with any other things. So dating for this group is very, very difficult. But they are much younger than the split of South America from Africa. Much, much younger. Maybe they evolved when both continents were still closer together. But these bidirectional transatlantic crossing, it's very, very common in plants. It happened 111 times. This was already found out in 1973. There was a fantastic paper about these junctions. It's all dispersed by usually water.
Yeah. Just think of it, millions and millions of years. I mean, these plants produce a lot of seeds.
This can be made very, very quickly. There's these floating mats of water plants and like a float, and you think it takes ages to cross the Atlantic, but in both directions it only takes 2 weeks. So,
and remember, we have coastal habitats in Brazil where we have found Genlisea growing. Yeah. You know, so maybe from one coastal habitat to another.
And if monkeys can make it from South America to Africa, then these tiny seeds, the little plants, they can make it as well. Thank you.
Is this you?
No, that's Andreas. That's my co-author.
Personal statement there.
Yeah. Question back there.
What's the current knowledge about the prey? Because there's mentioned nematodes, protozoas. Are there ones or second or both and many others?
I confess I have a paper in the making with Studichka where we actually opened up a lot of traps in Genlisea aurea, and I have some pictures if anybody wants to look at them afterwards. There's a lot of stuff in there. Some of them alive, swimming around like mad. I don't know if they will be dead or if they're part of that commensal community that lives within the traps. I mean, the main focus of this presentation, what I wanted to show you guys, is how little we know of this group. We really don't know what's going on in there yet, in these traps. Yeah, so what exactly do they trap? You know, and like Studichka saw with the 2 different traps, are the short ones near the surface capturing one sort of prey and the bottom ones capturing another sort of prey? Are they actively attracting things, or do things just go in there because there are little holes in the soil that they want to, you know, oxygen rich?
So, for those people, more practice related, for those people who grow Genlisea, especially Margarita and Genlisea aurea, propagated in high numbers. If you have it in vitro, produce tons of it. Because I get so many requests because scientists who don't know about carnivorous plants, they want to research with Genlisea. Because, as Fernando already said, for us, these 1.3 million base pairs, well, consider this as a book, a user manual for the human being, and it's written on 1,300 pages. And somehow, for Genlisea, which is also as well complex, this user manual is very, very tiny, just a few pages long, so it's easy to read. So everyone wants to work with Genlisea now, all the scientists. And the good thing for those of you who grow these plants, want to sell these plants, they need high numbers, huge numbers for this sequencing. I think 10 grams of Genlisea per study. So you can get rich if you propagate Genlisea aurea or Margarita.
By the way, Kamil, I don't know if you know, but those Genlisea aurea are from you.
Most of them are from you.
Yeah.
But make sure you've got your CITES permit.
No.
The good thing Genlisea is not on CITES.
Yeah, so if anybody... Oh yeah, so wait, one last thing about this focus of the talk. So, you know, in ex situ preservation, it's sort of, I don't know if this was clear, but this is where I wanted to get at. Without these plants in cultivation, we wouldn't know half this stuff. Notice how many publications we had in the last 10, 20 years, only because we got these plants into cultivation, or else we wouldn't know all this stuff. And how much more are we going to find out thanks to the fact that we got these plants into cultivation, and that all of you are growing this stuff and selling them and making them available to scientists who are discovering all these amazing things with these cultivated plants. So not only the preservation of them, you know, for scientific work in ex situ. Endangered wise, yeah, there are some species of Genlisea which are only known from a few locations. You know, like that Itacambira one. Maybe it's more widespread, we don't know. We've only found it at one little location. Some of those African ones, maybe they're more widespread, but they're in very remote areas. Maybe we'll find more population. But some of these species are only really known from one location. Hopefully not endangered yet. Maybe in the future, hopefully not. But yeah, so the preservation of them in cultivation is extremely important, not only for these small populations in the wild, but for this extremely important work that's being done with these plants by a lot of scientists. Any other questions? Well, thank you very much, everyone.